Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science Podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com. And this week, I am so delighted to introduce our special guest on the show. With us, we have Dr. Peter Vanderklish, who is the Chief Scientific Officer, at Spinogenix, which is a company developing therapeutics designed to restore synaptic connections, targeting neurological conditions such as ALS, Alzheimer's disease, schizophrenia, and Fragile X syndrome. Dr. Vanderklish has studied synaptic connections and their molecular biology since his graduate work at UC Irvine, where he helped show that long-term potentiation occurs post-synaptically in mature dendritic spines. He also identified integrins as key players in extracellular matrix changes that support lasting synaptic restructuring. So really key developments and findings and discoveries, indeed, that Dr. Vanderklish has undertaken and pursued and come upon and leading up to Spinogenix. And so in recent years, Dr. Vanderklish has focused on dendritic spines and synaptic connections in Fragile X syndrome, studying the molecular, synaptic, and neural networks to correlate them with the chronic anxiety and social avoidance behaviors of patients with the disease. Dr. Vanderklish has been an associate professor in neuroscience at the Scripps Research Institute and a visiting professor in anatomy and neurobiology at UC Irvine. So to learn more about the science behind Spinogenix and the diseases indications that the company is targeting through its innovative approaches, let's turn to Dr. Vanderklish now. Thank you so much for being on the show with us today.
Thanks for having me. It's a pleasure.
So Dr. Vanderklish, as I mentioned, really fundamental, foundational work that you've done in the field. And so I'd like to learn a bit more about that. What got you into the field? What piqued your interest? And how has that foundation really led your work now up until and to Spinogenix?
What drew me into the field? I'd say, like a lot of things in science, a little bit of luck. I was an undergraduate at UC Irvine and really didn't have firm ideas on what I wanted to do, but I did start doing some undergraduate research in the laboratory of Gary Lynch, a very colorful and genius, I would say, neuroscientist in the neuroplasticity world. And I got quickly shunted to projects that were studying what I think remains one of the more intriguing and challenging topics in neuronal cell biology, which is how does a synapse, in particular a glutamatergic synapse, change its efficacy state, increase its strength, within an instant in response to specific patterns of synaptic activity, and then how does it make that persist for very long periods of time, sometimes a lifetime of the animal. And so we started looking at the underlying biology of it all. How is it induced? What are the triggers at a molecular level? How is it expressed? You mentioned the locus of expression. Post-synaptic. This is something Gary's lab was very much involved in at the time. And then you're looking at the molecular and structural changes that have to happen to consolidate the functional changes. And so all of that was really fascinating and drew me into synaptic neurobiology. And then while I was in the lab, we also did some work on neurodegenerative diseases, including Alzheimer's. And it got me interested in exploring synaptic neurobiology as a way to find new ways to treat these diseases. There was a rather poetic hypothesis in the lab at the time that Gary had, which was these mechanisms of memory formation, this ability to have synaptic plasticity occur rapidly and over the long term, involves mechanisms that are so much to share to some degree with neurodegenerative diseases. And what he called it was, the price of memory. The fact that you have this extraordinary plasticity at glutamatergic synapses also comes with a price, and that is that synapses and neurons are particularly vulnerable to all kinds of stresses, and this is a component to neurodegeneration. And I think that still has, holds some water today. But that's what got me started, was working out that.
Wonderful. Thank you so much for sharing your journey and all of that foundational work that has now led you to Spinogenix. And so I'd love to learn a bit more about the company. And I know we've had a couple of other executives from Spinogenix on the show, but I'd love to hear your perspectives and what led you to join Spinogenix.
Well, it was fun. I had some. Uh, it was a little bit serendipitous, to be frank. I was at Scripps research, doing a lot of work, pursuing a lot of different projects. We were working on Fragile X syndrome. We were working on many other areas of synaptic neurobiology at the time, and it was during a period of particularly challenging funding environment. And I was aware of what was going on at Spinogenix, and it really just fit the bill of what I wanted to try to participate in terms of looking at the synaptic neurobiology of neurodegenerative diseases and neurodevelopmental disorders and Fragile X. And I kind of jumped ship, as many do, from academia to biotech, and it was an easy decision when the opportunity arose, because it really was a chance to, I think, have um, a very proximal and hopefully profound impact in many areas.
And how was that transition from academia to biotech? I think the environments are obviously very different. Sometimes the goals are different, but we're also at the same time seeing a lot of collaborations, increasingly more and more so collaborations between different industries and different areas, including academia and industry. And so how was that transition, and what are, I guess, the pros that you can take from academia and translate perhaps better in industry, and what are some of the pros and cons, I guess, in both areas, and how has that transition been overall?
The transition was, intimidating at first, to be frank. But it ended up being easier, than I thought. And one of the things you mentioned, was one of the reasons anyway, and that is that there's a lot of dynamic these days between biotech and academic centers. We, in our development of synaptic regenerative small molecules and BK channel positive modulators, we've routinely engaged academic experts to help us de-risk things going forward. So there is that. I actually found the funding environment to be a little easier overall. I think you become aware when you go from academia to biotech of many more options for securing the funding necessary to move things forward. And I think we found success there, and so that helped with building some confidence in the area. And then there's a number of things I think you never really leave academia entirely. You bring some lessons going into biotech, and I think some of those have helped. I think one of them is you always have to stay curious. In biotech, it's easy to get channeled, and along a particular development path. You have an asset, you have an indication, you know what the next step is, and it's easy just to kind of tunnel vision there. And I think staying curious, and thinking about things in different ways, different perspectives, even for well progressed assets, it helps move the needle in drug development. I also think that the value of multidisciplinary research, that's done so much in academia, carries forward very well in biotech. It's very important to bring various domains together at once, and that helps move things more concretely and faster. And then there's something that I kind of saw a lot at UC Irvine in Gary Lynch's lab, and was reinforced at Scripps, is when you're in academia, sometimes you realize that the best way to test your hypothesis. The screen went blank. Sorry. When you're in academia, sometimes you realize that the best way to to pressure test your hypotheses is through, I guess for lack of a better word, triangulation. You've got to formulate your ideas in as many ways and as diversely as possible. And again, this is something maybe it's easy to forget in biotech. As you have assets that's going along from step A to step D in a particular indication. You need to think about, what are the expectations, the testable predictions for your ideas in different contexts. How would it be in context A or B, and does that support your idea? And in biotech, that has the very useful translation that it helps everybody if you basically try early on to kill your own technology. That may seem weird, but you really need to put it through its paces. It helps everybody. It helps you if you want to make an impact. You don't want to waste any time. It helps your team. It helps investors. It helps everybody, especially in the end, patients who really are the heroes of clinical development. You don't want them wasting their time with something you're not, very confident about. So I guess that's one thing I cared for from academia to biotech. Try to give your technology an opportunity to fail as much as possible early on. And our CEO is of the same mindset. We've done that with our synaptic regenerative compound in spades, testing in different labs, different hands, different models with different outcomes. And it just keeps on, passing the test. And in our clinical trials, we've taken that approach as well. We've baked in many different objective quantitative outcome measures, including neurophysiological outcomes that really put it to the test, along with behavioral measures. And so far so good. We've seen some signs that it may be translating as hoped to the human conditions.
Yeah, no, I love that, having that really, not to say casting a wide net, but really trying to cover all the bases and to really test your hypothesis in various scenarios, and to really come upon something that's robust. And I really do love that approach, and definitely things that we, have in academia, learning grad school, of course, learning from all of the failures and setbacks is so key. And in terms of Spinogenix, I know you mentioned one of your compounds, and I'd love to learn a bit more about the pipeline at Spinogenix. And again, you don't have to go into any specific details about specific compounds and things like that, but just like to get an overview of really the approach that Spinogenix is taking to tackling some of these diseases, these indications, whether it's Alzheimer's, Fragile X, of course. And so I'd love to know really the mindset and approach that Spinogenix takes to developing its pipeline.
Yeah, so I think the overarching theme is that we're focused on the synapse. And it's in general, we think of it as synaptic restoration. And so in the neurodegenerative diseases, Alzheimer's, ALS, and schizophrenia, all of which we're in clinical development for, this entails use of a synaptic regenerative compound, because all of these conditions, these neurodegenerative diseases, and many others that collectively are known as the synaptopathies, involve the loss of glutamatergic synapses as a very early and progressive step in pathogenesis that gives rise to symptoms. And so our strategy there is to regenerate those synapses. And this is not a new idea. In Alzheimer's disease, where the prototypical synaptopathy was proposed back in the 80s, late 80s, early 90s, that any effective therapy going forward would have to stop synapse loss, or even better, reverse it if there were to be any hope of recovering some symptoms or some function, I should say. But the pharmacology just wasn't available. And we think, our lead candidate in that area has the chance to offer that therapeutic angle. So there's that. Now, in neurodevelopmental disorders, starting with Fragile X syndrome, our synaptic restorative approach is to correct, if you will, very specific synaptic dysfunctions that occur in the syndrome. And in this case, we're we're going after deficits in a potassium channel called the BK potassium channel that plays a big role in modulating synaptic activity and neural excitability.
And in terms of the clinical development pathways of some of the compounds in your pipeline, what is the approach to that in terms of trial design? And, we're seeing a lot of different quote unquote trends, whether it's patient centricity, diversity. However, I think it's really important to look at them as beyond just trends, of course. And so I know that to really incorporate these organically at every point in trial design. And so what is Spinogenix's approach? And especially in diseases that it's interesting because I feel like you're in a space where you're looking at diseases that are more common, like Alzheimer's, but then also rare diseases like Fragile X. And so what really are the different approaches potentially when you're looking or recruiting for trials for something like for Alzheimer's versus a Fragile X? I can imagine different approaches potentially in terms of just like patient population sizes and so many more considerations. I feel like I'm asking like a bunch of questions in one, but it's just so interesting to me. So I'd just like to know, I guess, from a clinical design perspective.
Yeah, I see where you're going with that. And that's a great question. So it's, trial design is a very evolving, iterative process, as far as I can tell, as a newbie in the biotech field still, I think. And our strategy in terms of recruitment, it differs from the neurodegenerative diseases where we're using a synaptic regenerative therapy to Fragile X syndrome where we're using the BK channel opener. And so I guess I'll start with the latter first. So in Fragile X syndrome, our phase 2A sort of proof of mechanism, proof of concept, go, no-go trial really involved a very selective subgroup of subjects. And those were what's called full mutation males, which have very little or trace amounts of the protein encoded by the gene that's silenced in Fragile X syndrome, FMRP, and they are the most strongly impaired. They also happen to have the greatest expression of neurophysiological changes that you can measure by EEG as a quantitative objective neurophysiological outcome. And so we use that as our sort of anchor point for first proof of concept, exploiting homogeneity in the phenotype to tease out an effect that we then think could be generalized further across the broader population. And in neurodegenerative disorders, it's an inclusion criteria, of course. It's such a big topic. Of course, it changes with phase. Right now we've been very sort of liberal in terms of our inclusion criteria. We're guided by KOLs in the field, clinicians, the PIs in the trials in terms of now, where our best focus should be. But in general, it's been pretty broad and defined by specific thresholds within key functional outcome measures and so forth. And then in terms of the overall design, one thing I think that differentiates Spinogenix from many others in the field, past and present, is that we are utilizing across all of our trials, all of our phase 2 studies, and we think going forward the larger pivotal trials. We're using a combination of behavioral outcomes, functional metrics, be it motor function in ALS or memory, for instance, in Alzheimer's, various metrics in schizophrenia. We're combining those with a number of different biomarker approaches. And the one that's really unique here, I think, is the incorporation of again, quantitative, objective neurophysiological outcomes across all trials. In Fragile X, that means monitoring signature changes in gamma band activity and the EEG, which Craig Erickson, who was on your podcast before, described very elegantly, and he's been a pioneer in this field. In ALS, it's also using EEG to look at signature changes in brain activity patterns that are correlated with symptoms and are thought to contribute to symptoms to see if we can actually normalize brain activity. And then it also within ALS, we're using transcranial magnetic stimulation, which gets at cortical hyperexcitability, a prominent early feature of ALS pathogenesis that contributes to further progression and informs on prognosis. And then in Alzheimer's and schizophrenia, we're using similar approaches, quantitative electrophysiological readouts that are sensitive to synaptic function, but which have also been used to define very specific abnormalities in brain activity that are linked to disease progression.
And absolutely, that's very important. I think you mentioned the various types of biomarkers, and we've seen definitely the field having evolved really towards incorporating more biomarker, dare I say, led development in neurobiology and neurodegenerative diseases. So I think that's absolutely very much the thing right now. And how do you think the field has evolved, and how important are biomarkers? And you mentioned using in conjunction, of course, having them as quantitative measures. Do you see the field really taking biomarkers further, or are there other technologies, other elements that are coming about that you see in the next couple of years will really help move the field forward?
Yeah, I think biomarker development is a very vibrant area of innovation right now in the space. I think there's a lot of great work being done. There's still obviously a lot of progress to be made. Some particularly exciting examples I think are come from ALS, where there's the potential for things. Everybody in ALS wants to be able to have some kind of method to measure the most common molecular pathology, mislocalization of TDP-43 to the cytoplasm. And but it's clear that's involved in pathogenesis. It's a modal sort of feature of the molecular pathology of the disease, and it's, by the way, contributing to synapse loss in a strong way. But nobody can measure it yet, non-invasively, and so everybody's trying to find a way, because that would really help guide therapeutic development, clinical trial enrollment. It can be used as an outcome measure, et cetera, to look at disease modification. And so, one of the consequences of this mislocalization is, of course, to change RNA splicing, and you get the inclusion of cryptic exons within certain transcripts, and these turn into sometimes they're translated to peptides, and people are trying to find either the evidence of the translated peptides or neoepitopes in plasma, or maybe even cell-free DNA or RNA, for instance. And I think there's a lot of potential there for detecting the disease process early, which would allow intervention trials as prevention trials, and maybe other kinds of trial designs, and maybe even trying to assess efficacy very early in a patient population that really, frankly, just doesn't have a lot of time. So if you can figure out that an intervention, a therapy is failing, you would know it soon. You can let them try something else. If you see it's working, well, fantastic. And then there are other approaches, to try to look at TDP-43 and other tissues. One of those is actually, of all places, the retina and the eye. There are methods in development now for being able to non-invasively label TDP-43 aggregates and visualize those in the retina. So that's a good example. But yeah, it's a very vibrant space. Everybody's waiting for the next level of innovation because it always impacts trial design and capabilities and things. So, I think there's a lot coming that will be of strong value.
Indeed. And for Spinogenix, in terms of the company's pipeline, is the company looking to develop its candidates as monotherapies or in conjunction with other approaches and therapies? Because I think this is such an innovative, but as you also mentioned, not so new, but it's an intuitive also innovative because not a lot of companies, not a lot of teams are focused on the synaptic loss aspect in a lot of these diseases. And so would the idea be to solely target synaptic loss, or would it be potentially in conjunction with targeting things like beta amyloid, which is the classic hallmark of Alzheimer's, let's say. But as we know, we've seen with some of the monoclonal antibodies, while they've been able to lower beta amyloid, it hasn't translated necessarily into clinical efficacy. So do you think something like targeting synaptic loss could potentiate and improve the clinical efficacy of those kinds of approaches and vice versa? Or do you think targeting synaptic loss really could be the one powerful hit that's needed?
That's a really good question. And I think the answer, of course, is going to be different for different diseases. I think, you touched on some very important issues. Going forward, I think, yes, absolutely, synaptic regeneration, be it with SPG302, our compound, and maybe other neuroplastogens, as they call them, that are being developed, is going to be a mainstay in the treatment of neurodegenerative diseases. I mean, everywhere you look in the neurodegenerative space, you have synapse loss, and in some cases, it's a very primary contributor to symptoms, and others it's a little bit more distal. So you may get varying degrees of benefit from regenerating synapses, but you probably will get benefit almost universally, would be my prediction. So it's going to be a mainstay. In some, it could be administered as a monotherapy and have considerable benefit. But I think really, eventually, I think you're going to see it being SPG302 and other neuroplastogens used as a regenerative component. So cocktail therapies that address underlying pathomechanisms at the molecular level. And an important feature of that too will be cocktail therapies that are personalized. As we learn more from biomarker development, how different patients within a particular indication vary in terms of the relative contributions of different pathogenic processes, you'll be able to design precision cocktail approaches that address that heterogeneity in terms of pathogenic mechanisms in a very specific way to slow them or arrest them. But you're still going to need a regenerative component to get connectivity back. I mean, lost connectivity is lost function. And if you don't get that connectivity back, you're unlikely to get much function back. And another important thing to consider too is I think in a lot of these neurodegenerative diseases, people think, what's the pathogenic process? What's the pathogenic process? And this is great. You need to define the underlying etiology of it. You need to define the heterogeneity of pathogenic processes that are involved. But what's missing in a lot of conversations, and which is the reason we're doing what we're doing, is the conversation that was mentioned about regeneration. Many people are thinking about regenerating synapses, although it's growing in recognition. And an important component to a lot of these neurodegenerative diseases, especially the aging component, is that you not only have pathogenic processes such as overzealous microglia eliminating synapses, but you have on top of that a failure of regenerative responses of neurons that through most of a patient's lifetime have offset the consequences of the pathogenic mechanisms. In terms of where the rubber really hits the road in cognition and memory, synaptic density. And so I think there's a great potential for SPG302 and other neuroplastogens to sort of reawaken that regenerative process in these diseases. And that will be a very important complement to the sort of precision biomarker-informed cocktail therapies addressing underlying pathogenesis.
It certainly seems very, very promising, and absolutely, the work that you're heading and helping lead at Spinogenix is a part of that promise. So, Dr. Vanderklish, thank you so much for your time today, your insights, sharing, the very important work that you're leading at Spinogenix. And thank you so much for all your work and for your time with us today. Thank you.
Oh, thank you. Thanks for having me.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next time. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.